in the last recent years. So yes, I will present a hypothesis on the mosaic evolutionary history of brain circuits, how some circuits evolved in a different manner to others. And just to jump on the question, you know that we work in the lab with different species. We are interested, let me see whether I can find the laser point. Yeah, let me see, not yet. Yes. Yeah, now, so we know that all the different species of vertebrates that we studied, they have homologous regions. So for instance, we have a sagittal section here of the mouse brain, equivalent sagittal section of the gecko brain here. Both species, they have a hypothalamus. Both species, they have a thalamus here, or a optic tectum and superior colliculus. These are homologous regions, but each of them, in these homologous regions, they generate different types of cells and also different types of circuits. And we are interested in understanding how these structures diversify in evolution. For this, we work with 3 species in the lab. We have the mouse as a representative species of mammalian brains. We have the gecko that I will present to you soon as a non-avian reptilian species, and also the chick, which is our bird species that we work in the lab. But also, we have to understand that evolution is best understood from the developmental point of view. So we work with this hypothesis in the lab coming from the developmental biology field. We think that early on in development, when the first neurons are appearing in the brain, in all different vertebrate species, there is a common brain form, there is a common scheme in all these vertebrates' brains. And this is conserved, we call it the phylotypic brain, because it's conserved throughout the whole phylum of vertebrates. And from this common template, which will show, we hypothesize equivalent and homologous cells, equivalent and conserved genetic programs, etc. From this, each species will derive different types of brain according to the different types of developmental programs, neurogenesis, cellular migration, differentiation programs, etc. So we think, I don't know if you can see here the whole slide. Let me see stuff here. Anyway, so we think that there is a common brain scheme, homologous cells and conserved genetic program in these phylotypic brains that we will see in all different vertebrate brains, but also that this common brain will have an impact on how the different brains will develop further on. So there will be preserved rules of neurogenesis throughout development. Please let me know whether you can see the right side of my slides. Is it okay? Can you see it?
Yep, I can see it.
Okay, I see your faces there. I have no idea whether I'm pointing or not. Anyway, so these are the 2 main things that I will present to you today. First, whether there is a phylotypic brain or not, how much conserved is this early form of the brain in different vertebrates. And second, to how extent this phylotypic brain is impacting on the neurogenetic program in the different brains. For the first part, how much conserved is the phylotypic brain, we resort to do single-cell RNA sequencing. So we extract the phylotypic brain of different species. This is just one example of the chick brain, which is our preferred model in the lab. At very early stages, this is 3.2 days of incubation when organogenesis has ended in these animals. We dissect the brain out and we perform single-cell RNA sequencing. We have identified all the different structures and cellular types that we expected. Most of these are neural stem cells, cells belonging to secondary organizers of the brain, and also the earliest neurons in the brain. And also we've seen that they organize in the UMAP according to the different regions in the brain. So clusters belonging to the forebrain, some others belonging to midbrain, hindbrain, and also to the different neurons of the brain. So each UMAP that I will present to you today, they have a neuromeric organization. The neuromeres, or the rings of the neural tube, will be present in these UMAPs, and we will try to compare them. We are working now with 5 species. In all of them, we study the cells of the phylotypic brain stage, which is very early in development. I have presented to you earlier the chick data set that we are studying on. Also, we have a mouse data set derived from an article from Linus group in Stockholm. We have a zebrafish data set from equivalent stages of the phylotypic stages coming from Alex Shearer's lab. And these are the 3 main datasets that we are comparing. But beyond, we have our own gecko dataset. We are working on and analyzing it already, but this is too preliminary. I will not show you today. And also human cells that were sequenced and published by the group of Arno Christian in San Francisco. So we are comparing these 5 species at the very early stages of brain development, and what we've identified so far is a conserved general organization of brain regions and cell types. And these cell types belong to the 3 main types: stem cells of the different areas, earliest neurons of the brain, and also the secondary organizers. To compare them, we put the 3 data sets together into a single data set to see how much these cells are similar to each other according to this species. And we can see here the 3 maps from the 3 main species under comparison. And when we merge them together, the data set of the merge is showing a quite nice intermingle of cells, showing that these cells are quite similar to each other, and the main differences are according to brain areas and not to the species which the cells are coming from. Also, we have performed other types of analysis. This one is called the Gen Specificity Index Correlation, and in this case we compare the whole transcriptome or either only the transcriptome factors expressed in each of these clusters between two species. For instance, we have the chick data set here. These are all the clusters on different regions and cells that we identify, and we compare them to the data set of mouse. And in every case, what we see is that cells belonging to one type of cell, for instance, roof plate here in the chick, are most similar at transcriptomic level to the roof plate of the other species. This is a comparison that we've done chick to mouse, mouse to zebrafish to chick, etc. And in every case, we see the same. Cells are quite similar according to the area in which they are derived. The cells are highly similar at transcriptomic level. And a final comparison of this first study that I wanted to present to you today is performed with label transfer analysis. In this case, we have the UMAP of the chick, which we see in pale red below all these other bright cells. And then we interrogate all cells from a secondary species, in this case from the mouse. We interrogate each cell to what cell of the chick UMAP are you most similar to, and they decide, and they are distributed accordingly. And we have seen exactly the same results. So cells from an area in the mouse data set are most similar to cells in the other data set, in this case of the chick, showing that there is cells at the equivalent organizer. The cells, the neural stem cells in different regions are quite homologous. And also, in the end, that there actually is a phylotypic brain, which is highly conserved, not only at cell level, but also at transcriptomic level. So beyond this, we wanted to see, as I told you earlier, on a secondary stage, how much this early conservation of the phylotypic brain, how this conserved form of the phylotypic brain is impacting on the neurogenesis program in all these species. So we have moved to understand how neurogenesis is happening at many different levels in the 3 experimental species we have in the lab. As I told you, mouse, chick, and gecko. So the main experiment that we're doing is with birthdating. We inject EDU, which is an analog of the thymidine, and when there is DNA synthesis, this EDU molecule goes into the nucleus and it appears there. We can reveal it and find it later on to see what cells were dividing, for instance, 2 hours after the injection. In this case, all cells here in the chick embryo located next to the ventricle, of course, where mitosis is happening. But if we wait for 7 days, we will see where all these newborn neurons are located in the mature tissue. We are comparing different stages of injection, early in neurogenesis, intermediate stages, and late stages. And we are also identifying cells at different areas and in different species. This is one example. We have this Paradula pictus, the species of gecko coming from Madagascar in Africa that we are researching on in the lab. And this is an example of the injection. I don't know, we can play, maybe not, it's not working now. But anyway, we can see here in green, the injection of EDU within the ventricle. The size of the egg, just to let you know, is similar to the size of 1 cent of a euro coin. So it's smaller than 1 centimeter. So we perform the manipulations in these embryos there. And then we compare gradients of neurogenesis at many different levels. So for instance, we are researching whole brain gradients of neurogenesis across the entire brain, but we focus also on different brain regions such as the thalamus, hypothalamus, etc. We go and study gradients of neurogenesis in a specific nuclei and specific circuits, And finally, we identify the neurogenic time points at which specific cell types are generated. Briefly, I'll show you here how whole brain gradients are conserved across different species. This is a sagittal section of a mouse and equivalent stages in a chick. And in both cases, in gold, we have labeled with EDU the earliest neurons generated, or nearly the earliest neurons generated in the brain. And you can see that the location of these early neurons is quite similar in both species. So there is a higher amount of neurogenesis in caudal levels here at the rhombencephalon in both species, whereas in the telencephalon there is barely no neurogenesis apart from a few cells in the axexular olfactory valve. Both species also is not present here in the chick. And also we see that there is more neurogenesis at basal regions here below this alar basal lamina, and less neurogenesis or pure neurogenesis in alar domains, such as the thalamus here, prosomere 2, or in the superior colliculus in the most dorsal area here, in the optic tectum, and also here in the alar part of the hypothalamus. So equivalent gradients of neurogenesis across the entire brain is what we identify when we study different species. When we zoom in to different brain regions, this is an example of neurogenesis in the hypothalamus of the gecko. Injections at very early stages, E7 in the gecko, we see neurons in the hypothalamus locating mostly the most lateral sides. This is the lateral hypothalamus. And when we perform these injections, but later on, E9 or E11, etc., we see that the cells do not migrate readily that far to lateral portions of the hypothalamus, and they remain more in the intermediate regions of the paraventricular hypothalamus here, and also later on they remain attached to the ventricle. The extreme cases are E15, where only neurogenesis happens in the arcuate nucleus, and the cells are just next to the ventricle. So there is a lateral to medial gradient of neurogenesis in the gecko, and this is exactly the same gradient that we identify in the mouse, and it was identified by Allman and Bayer long ago. We can go deeper. We can go, instead of brain territories, we can go to a specific brain nuclei. And in this case, I'm presenting to you today the neurogenesis in the habenular complex of the gecko. We can identify this habenula. It appears at the dorsal-most area of the thalamus. And when we see when the neurons are generated, we see that the earliest neurons are generated locate the most lateral side of the habenula. And then later on, at different stages, they locate intermediate stages, most medial one, and the latest one, at E15, locate the most medial side of the habenula. We can collapse different animals into one single image, pseudocoloring the EDU labeling, and you can see the gradient from early magenta in the lateral habenula to cyan in the medial side of the habenula, the late neurons that are generated. And it is interesting that this lateral to medial gradient of neurogenesis, you can bet it is exactly the same gradient that it was described by Allman and Bayer in the neurogenesis of the rat habenula about 40 or 45 years ago. Early neurons go to the lateral habenula, whereas the latest one go to the medial. So this neurogenesis process is conserved also at the level of specific nuclei. And when we go to circuits, this is a beautiful example here in the cerebellum. An early injection in the cerebellum, here we can identify where the cerebellum is located in a sagittal section of the gecko, and we see the EDU labeled in white. Cells in the cerebellum are located in the Purkinje cell layer. So we can identify them here. These cells are expressing parvalbumin, do not express PAC6, therefore they are Purkinje cells. And what we know, we have repeated these experiments for many other stages in the gecko and also in the chick, and what we have identified is the exact same sequence of neurogenesis of the many different population of neurons in the cerebellum. First neurons in magenta are Purkinje cells, both in chick, in gecko, and also it was described for mouse. Later on, some granule cells and first GABAergic neurons on the internal granule layer. And finally, the most abundant population, which appears here in cyan in both species, are the granule cells, glutamatergic cells of the granule layer, and the latest GABAergic cells of the molecular layer, also here labeled in cyan. But it is remarkable that beyond over 320 million years of differences in evolution, the same sequence is conserved, the same generation of types of neurons is conserved among these three species of amniotes. So we can think that the phylotypic brain, which is appearing, as I told you before, appearing very early in development, has an impact on early neurogenesis, but we have tested what happens at the end of neurogenesis. Is there any conservation of the late events? And you can bet that there is. This is an injection at E27 in the gecko. This is comparable to a postnatal state in the mouse. This is very late. This is 2 weeks later, any neurons is generated, all neurons are generated already in the thalamus and the cerebellum, etc. And the 4 different niches in which we have identified neurogenesis so late in development, you can guess. These are, of course, the granular layer of the cerebellum, in the olfactory regions related to olfactory bulb, which is not appearing here. The olfactory bulb is quite distant from this area. It doesn't appear on the photograph, but neurons are generated there. Also, in the medial side of the cortex, in the medial cortex, this is homologous to the dentate gyrus of mouse. And also, finally, in the arcuate nucleus of the hypothalamus. And these 4 regions are the main 4 regions of late neurogenesis, late postnatal neurogenesis, or even adult neurogenesis in mice. So the same conservation of late neurogenesis is appearing here in the gecko brain. So far, I think I have shown you enough evidence to convince you that there is a huge amount of conservation of the neurogenic process coming from very early stages to very late, and happening from whole brain gradients to a specific nuclei and specific cell types. But of course, the title of the talk starts the same: mosaic evolutionary history. So not everything can be conserved. Some other territories have to escape to the rule. And this is the case that we have identified for the cells belonging to the sensory processing circuit in the cortex. Very briefly, we know that there are cells communicating in a specific manner in the neocortex of mammals. So this is a 3-synaptic circuitry. It means that there are 3 glutamatergic neurons communicating each other. Information arrives to them from the thalamus to layer 4 in the cortex. This is what we call the thalamic recipient neuron. This one communicates with upper layer neurons in layers 2 and 3. And these are integrators. They receive information from different glutamatergic neurons. Later on, they send a projection to deeper layer neurons in layer 5 and 6. And these are the ones that project out of the cortex or out of the pallium. So this was termed the canonical circuitry or the columnar circuitry in mammals, and it was thought to be specific of mammals, something like the achievement or the crown of evolutionary processing in the brain of mammals. But it was long ago, 50 years ago, that Harvey Carten in the States shown that there is an equivalent circuitry. First, he showed in pigeon, but later on in other birds and also in reptiles. There is an equivalent 3-synaptic circuitry in the pallium of other non-mammalian species. And he thought this circuitry has to be homologous because of the many similarities, functional, molecular, etc. We wanted to know whether these 2 circuits, the canonical circuits of mammals and this 3-synaptic circuit of chick, are homologous or not, following the same assumption that we did for the cerebellum. The sequence of neurogenesis should be conserved between the homologous cell types. So are these dorsal ventricular ridge in chick and neocortical circuits homologous? We know very well by the studies of many groups in the past when the different neurons of the neocortex are generated. What is the sequence of generation? Very briefly, there is a tight link between when in development you are generated and what type of neuron you are becoming in the cortex. Earliest neurons located in deeper layers of the cortex, and these are the ones that project out of the cortex. Later on, at the intermediate stages, we have layer 4 neurons, that act as thalamic recipient neurons of the circuitry. And the latest one generated in the pallium, in the cortex of mammals, are the integrators, here in blue, appearing in layers 2 and 3. So there is a tight sequence between when you are generated and what type of cell you are. We have researched this thoroughly in chick. This was a massive amount of work, injecting dozens of hundreds of animals in chick and also in gecko, although in gecko I'm not presenting it to you today because of time limitation. And this is an example. We have in white here in a coronal section of the chick brain, the location of cells generated at the intermediate stages of neurogenesis. And we confront this with different immunostainings, related these immunostainings to different markers for GABAergic cells or glutamatergic cells, etc. And we have counted thousands of cells to end up with this summary here. First neurons generated in the circuitry are appearing here in the endopallium. The names are not relevant, and they are marked here in red. And these neurons happen to be the thalamic recipient neurons. Also, some early neurons located in the arcopallium, and these ones project out of the circuitry. Later on in development, in green, we have the dorsal endopallial neurons, which are the integrators of the circuitry, and the remaining arcopallial cells projecting out. And the latest generated ones generated at EH in chick are not involved in the circuitry. So we have neurons generated in the pallium at these stages very late, but these neurons do not belong to the processing circuitry. And as a summary of this, what we've seen is a completely different sequence of neurogenesis comparing the two circuits. The main two differences are here. So earliest generated neurons in the mouse or in mammals locate here lie 5 and 6, and they project out. Whereas the earliest generated neurons in chick appear here, and they are thalamic recipient neurons. Of course, some of them are also projecting out. Later on, it's when the other types of neurons are generated, but there is a lack of neurogenesis related at the end of neurogenesis in the case of the chick, whereas at this equivalent time point in mouse, there is the generation of the integrator neurons happening or settling layer 2 and 3. So the sequence of neurogenesis is completely twisted. Not only that, the regions of the brain in which these circuits appear are different. This circuitry can be generated from one single progenitor versus the two, at least, different progenitors that are generating these different areas. So as a summary, the whole developmental program of these two circuitries is different, and the cells and the circuitries cannot be considered homologous. We wanted to know a bit more at the level of single cell and transcriptomic, what happened to the cells? Are they comparable or not? Although they are not playing the same role or located in the same circuitry, if they are generated at equivalent time points, are they transcriptomically similar? For this, we have developed this method in the lab, which we call Birthdating, and it comprises that we label cells at the specific time point, E4, for instance, very early in the chick, or E12, early in neurogenesis in the mouse. And then we leave the cells to survive in the animal, and we isolate it when the tissue is mature. We isolate the cells, and we perform single-cell RNA sequencing, E15 in the case of chick, or a P3 in the case of mouse. These are the UMAPs. You may see here Cajal cells. Julia was talking about earlier today. And we have analyzed the different glutamatergic and GABAergic cells which are early born in the pallium of these two species to see whether they are comparable of transcriptomic level or not. And you can guess, they are not. So when we integrate, for instance, these two data sets, what we see is that there is a nice integration of the two GABAergic cells coming from the two species. Of course, there are some differences. These are mainly chick cells, chick GABAergic cells, which are not similar to other mouse cells. But when we go to the glutamatergic side, there is a complete lack of intermingling. Some clusters are intermingling here or there, but the main bulk of cells, glutamatergic neurons, including Cajal cells, are not intermingling. This suggests that these cells are not transcriptionally similar. We have continued this analysis that I will present to you in the last minute here. We have tested what is the composition of this cluster. We've seen that glutamatergic cells are more species-specific than GABAergic cells. So they're not as similar as we expected at the beginning of the project. And when we compare transcriptionally the transcriptive factors that are expressed in glutamatergic cells between the two species, what we see is that in every case, GABAergic cells are more similar than glutamatergic cells from the two species. There's only one cluster, which is here, mouse GLUT4, which is similar to chick GLUT4, but beyond that, the main similarities are coming from the comparison of GABAergic cells. So if these cells are not that similar, we wanted to also know to what type of cortical cell are more similar to. So we compared, finally, this glutamatergic cluster coming from the early pallium of the chick to all the different transcription factor sets that are known to be expressed by different types of neurons in the cortex. These are, the name of the genes are coming from Paula Arlota's lab that has, who has researched this thoroughly. And what we see is that our clusters, our glutamatergic clusters, are not similar to any combination of transcription factors from any projection neuron in the cortex. So just summarizing, glutamatergic neurons of the pallium of mouse and chick are transcriptionally different. And for sure, this is a reflection of their different neurogenic process and their different developmental program. So to wrap up, to finish with my talk today, I wanted to summarize the three main topics that I presented to you today. First, that we have identified a common phylotypic brain, that it exists in the early embryo of amniotes and also of the different vertebrates up to zebrafish that we have researched. And this is supported by a whole transcriptome analysis at single cell level. Second, that this common embryonic brain, this common phylotypic brain, imposes constrained rules of neurogenesis to all amniotes that we have researched. And these neurogenic rules are conserved from whole brain level to specific brain areas, to circuits, and also to cell type levels. But still, although there is a vast conservation of neurogenesis, there are still at least one region or one circuitry in which evolution has tinkered a bit and has found another way to escape. So beyond circuit conservation, some circuits have escaped the rule and have evolved separately in independent lineages of vertebrates. And this is promoting brain diversity. Just coming up back to the title, I just wanted to tell you that there are many different cell types and circuitries which are conserved, are built in a conserved manner that I resemble here as a pyramid, but still, at least there is one circuitry in the neocortex and the dorsal cortex and dorsal pallium of other species in which innovation of evolution has played a significant role. Just to finish, I would like to thank all my collaborators, the funding agencies that are supporting our research, but definitely and mostly Eneritz Redalaña and Rodrigo Seno Villaganzo, who are doing the PhD in the lab and are performing absolutely every single experiment and analysis, and they are awesome. So thank you very much to them and to all you for your attention. Happy to take any questions.